This is Dr. Mitch Robinson, and this video is on oxidative damage and cellular defense, and it accompanies handout number 12 from Cellular and Molecular Medicine. In this video, we'll examine the reactive oxygen and nitrogen species that cause damage to tissues, and we'll look at mechanisms that are used to defend against this damage. As we've seen, oxygen is a very powerful acceptor of electrons. We can derive energy from the transfer of electrons from donor organic molecules to molecular oxygen. But the two unpaired electrons of diatomic oxygen make it particularly susceptible to radical formation. If oxygen accepts less than the full complement of 4 electrons, then oxygen radicals are formed. Oxygen radicals are formed as a natural byproduct of normal metabolism, and these are known as reactive oxygen species. Nitrogen can form radicals as well, and these are known as reactive nitrogen species. There are also reactive species of oxygen and nitrogen that are formed exogenously, and these can be damaging to tissues as well. When reactive oxygen species increase in the body, they can cause damage to tissues, and this is known as oxidant stress. We have several mechanisms to protect the tissues against damage by reactive oxygen species, and we'll talk about these in a minute. Now, the figure on page 2 shows the reactive oxygen species. The superoxide anion is formed by the addition of a single electron to an oxygen molecule. This molecule is often formed in the electron transport chain when oxygen is incompletely and prematurely reduced. Superoxide itself is not particularly reactive, but it can interact with certain enzymes to inactivate them, and it can initiate lipid peroxidation. A peroxide radical is formed when superoxide gains an additional electron. The protonated form is hydrogen peroxide. The addition of an electron to hydrogen peroxide forms water and a hydroxyl radical. The hydroxyl radical can also be formed by ionizing radiation. This is an extremely reactive species that will remove an electron from almost any molecule it encounters. Reactive nitrogen species are formed both exogenously and endogenously. Nitric oxide is an oxygen-containing nitrogen radical that serves as a neurotransmitter and hormone. It's formed from arginine by the enzyme nitric oxide synthase. Nitric oxide is a potent vasodilator. Nitroglycerin breaks down to form nitric oxide in the blood, and it's given for ischemic heart pain or angina. You'll learn more about nitric oxide later in the course. Nitric oxide is formed in the vasculature and can interact with superoxide anion to form peroxynitrite. Peroxynitrite is itself a very reactive molecule, and it can react with lipids, amino acids, DNA, and protein molecules in the cell. Nitrite and nitrogen dioxide are exogenously formed reactive nitrogen species. Nitrite's used to cure meat, and it can also be formed by intestinal flora. Nitrite can interact with amino acids to form nitrosamines, and these are carcinogenic. Nitrogen dioxide is found in polluted air and cigarette smoke. Exposure to this compound is damaging to the lung. Now let's look at some sources for the formation of oxygen and nitrogen species in the body. The electron transport chain produces reactive oxygen species when oxygen is incompletely reduced to water at complex 4, or when electrons prematurely interact with water earlier in the chain. Now, there are many enzymes that utilize oxygen. These are known as oxidases, peroxidases, and oxygenases. Oxygen radicals are formed when these enzymes release oxygen prematurely before it's completely reduced. Now, a major source for the production of reactive species is the production during inflammation. Certain immune cells produce reactive oxygen species in response to foreign agents such as bacteria or fungi. Neutrophils catalyze the transfer of an electron from NADPH to oxygen to form a superoxide anion. The enzyme is NADPH oxidase. So in order to combat infection, neutrophils use other enzymes to form reactive products from superoxide. One of these is superoxide dismutase. Superoxide dismutase converts two superoxide molecules into hydrogen peroxide and oxygen. Now, hydrogen peroxide is not by itself highly antimicrobial, but it can be converted into a powerful toxin, hypochlorous acid, by the enzyme myeloperoxidase. So, phagocytic cells such as neutrophils and macrophages use respiratory burst enzymes, NADPH oxidase and myeloperoxidase, to produce antimicrobial reactive species. And this occurs in phagocytic vesicles within the immune cells. Here, the invading bacteria or other pathogen is killed by the generation of these toxic compounds. But these toxic compounds can diffuse out of the cells and do damage to the surrounding tissues, and thus their response to foreign bacteria can often lead to damaging inflammation. Free radicals from external sources, such as smog or cigarettes, or from endogenous sources, such as inflammation and the electron transport chain, can cause damage to several types of biological molecules. This includes proteins, membrane lipids, DNA. Damage to membrane lipids leads to lipid peroxidation, and this leads to increased membrane permeability. Page 6 is a diagram showing the formation of lipid peroxides from exposure to superoxide. A lipid peroxyl radical can react with another lipid to form another peroxyl radical. So this means the process is self-propagating, so it forms a chain reaction. Free radicals can react with DNA to produce strand breaks and damage to the deoxyribose backbone, and this can lead to cancer. Certain amino acids are susceptible to attack by free radical. This leads to the inactivation of enzymes. So there are a long list of diseases and conditions that have been associated with free radical damage, and this includes aging, cancer, arthritis, diabetes, kidney disease, lung diseases such as emphysema and bronchitis, and neurodegenerative diseases such as Parkinson's disease and Alzheimer's disease. Now let's look at some mechanisms by which we defend against oxidative damage. Glutathione peroxidase is a key enzyme in the elimination of harmful peroxides. This enzyme requires selenium for activity. Glutathione peroxidase uses the reducing power of glutathione to convert hydrogen peroxide to water. Now recall that the pentose phosphate pathway provides reduced NADPH to maintain reduced glutathione. Superoxide dismutase, which converts superoxide to hydrogen peroxide, is part of the respiratory burst in phagocytic cells, but it's also present in mitochondria and cytosol of other cells, where it serves as a defense against reactive species by converting superoxide to less reactive hydrogen peroxide. Catalase converts hydrogen peroxide to water and oxygen, so superoxide dismutase and catalase work together to remove toxic superoxide. And there are several non-enzymatic defenses against oxidative damage. Vitamin E is the general term for tocopherols and tocotrienols. Vitamin E is a lipid-soluble antioxidant. It protects membranes from oxidation by reacting with lipid radicals produced in the lipid peroxide chain reaction. We need vitamin E to protect against oxidative damage, but most studies indicate there's no clear advantage to supplementing your diet with extra vitamin E. Vitamin C functions as a free radical defense in addition to its role in collagen synthesis and other reactions. Carotenoids, including beta-carotene and related compounds, also act as antioxidants. Flavonoids are a group of polyphenolic compounds with similar structures, and these act as antioxidants. They occur in wine, tea, and other plant-derived foods. It's not clear how flavonoids exert their antioxidant effects. A third mechanism of defense against oxidation is the cytochrome P450 system. This is a large, diverse family of hemoproteins that take part in many important pathways. They're usually part of multi-component electron transfer chain systems called P450 systems. The term P450 derived from the peak wavelength for the absorption maximum for the enzyme, 450 nanometers, when it's in the reduced state. The term now refers to the group of proteins that contain this particular heme cofactor. Cytochrome P450, or CYPs, are found both in mitochondria and endoplasmic reticulum, and they occur in most tissues of the body. They carry out hundreds of different reactions, with the most common being monooxygenase reactions. Now, in a monooxygenase reaction, one oxygen atom is transferred to an organic substrate, and the other is reduced to water with NADPH serving as a source of hydrogens. CYPs play an important role in hormone synthesis and breakdown, including the synthesis and metabolism of estrogen, testosterone, cholesterol, and vitamin D. The P450 enzymes also function to metabolize potentially toxic compounds, including many drugs. Humans have 57 P450 genes, and these are organized into 18 families with 44 subfamilies. Many drugs may increase or decrease the activity of various CYP isoenzymes, either by inducing the biosynthesis of an isozyme or by directly inhibiting the activity of the CYP enzyme. This is a major source of adverse drug interactions, since changes in the CYP enzyme activity may affect the metabolism and clearance of different drugs. For example, if one drug inhibits the CYP-mediated metabolism of another drug, the second drug may accumulate within the body to toxic levels. Hence, these drug interactions may necessitate dosage adjustment or choosing drugs that do not interfere with the CYP system. The expression of different cytochrome P450 isoforms varies significantly among individuals, and determining the individual's genetic profile for CYPs is a valuable tool in determining drug dosage and interactions. Pharmacogenomics, or the genetics of drug responses and interactions, is a new expanding field that is increasingly important in medicine. And that completes our video on oxidative damage and cellular defense.